Welcome to today's lesson of generating proteins within the endoplasmic reticulum, specifically the rough part. First off, we have our mRNA that is generated in the nucleus, and it's connected to a ribosome. And whoo, look, we started making some proteins, but it stopped because the signal sequence is really important and it slows it down. And oh, psych! We have the signal receptor protein that bonds to it, which slows everything down. And then, oh, we met the signal receptor protein receptor. And then they kind of pull away as the whole thing is connected to the translocon. And then as it keeps starting to make more protein, the translocon plug goes away and we have more protein. Yeah, for protein. And then what happens is the signal peptidase comes over and cleaves off the signal sequence. Isn't that great?
It's amazing.
And then, whoa, that's a lot of protein. And once we ran out of mRNA, the protein comes through and we're looking pretty good. Then the dolichol phosphate, which has the sugar stored on it, is transferred by the oligosaccharide like so. Also, this already happened, but the translocon plug goes back, so FYI. And then the protein disulfide isomerase helps it fold up so it looks like this. That's pretty cool. And then after it goes through the ER working some magic, we end up with 2 less glucose. That's pretty neat. Additionally, the ER chaperone, known as calnexin, comes through and it takes off. Are we filming? Oh, okay. And then we take a trip after the calnexin, and we take a trip to the Golgi. Wow, we're here. This is pretty neat. And then in the Golgi, as such, a lot of sugar stuff happens, and we end up like this. That's pretty cool.
Yeah.
And then we take a ride in another transport vesicle, which looks almost like the last one, except now we have a RAB. And we take a trip, and we go back to another membrane for the outside of the cell. And now this is the outside and this is the inside. No, that's the inside, outside, inside. And we go, no, inside, outside. And we come in, and there's a RAB protein over to here, which is great. Same with the T-snare. And we have initial tethering occur between the RAB proteins. And it kind of brings it in like such, like parallel parking almost. And then the snare takes over. And as we go through here, we do fusion, and then bam, we're on the other side. And that's how you make proteins.